Today is our weekly webinar, and we were really excited to have guest speaker and team with us tonight. So if you can go to the next slide, we are really excited to have Dr. Cho, Head of Clinical Science at Calico Labs. As you know, for those of you who are following this webinar weekly, Calico Labs is the drug manufacturer working with us for Regimen F. We're really excited that we've been providing updates about this regimen. We had the previous webinar where Dr. Cho and his colleagues have discussed the science behind this regimen. And tonight, Dr. Cho is joined by a few of his colleagues. So without further ado, I'll let him introduce himself and the Calico Labs team. Bill?
Thanks, Sabrina. Yeah, hi, my name is Bill Cho, and I'm joined by a couple of other folks here from Calico, Amos Baruch, who is our head of biomarker development at Calico. And then we also have Carmela Sidorowski, who is our head of research, and Emmanuel Bellarmine, who is head of regulatory affairs and quality assurance. So maybe if you can go to the next slide. All right, so thanks for having me back. What I thought I would do over the next few minutes is to provide a refresher about our program, ABBV CLS 7262, that's in development for ALS, and we're proud that we're currently part of the HEALEY Platform Trial, currently enrolling as Regimen F. Next slide, please. So just to remind you about who Calico is, we are a small to medium-sized biotech company that was founded in 2013 by Google. And we have a bold mission, which is to understand human aging and to develop therapies for age-related disorders, including disorders that affect the brain. And since our foundation in 2013, we've been in a long-standing partnership with AbbVie, who is a great partner since they're a global biopharmaceutical company, and they have a proven track record for successfully developing drugs for various disorders of the brain. Next slide, please. So together, we've been interested in many fundamental biological processes, and one of the processes that we've become very interested in is something called the integrated stress response, or the ISR. We believe that this pathway is intimately involved in the cause of ALS. Next slide, please. So I just wanted to quickly remind everybody about the integrated stress response, or the ISR. This is at the highest level, it's a fundamental biological process that allows all the cells in the living organism to respond to various types of biological stress. And the two key players in this pathway are enzymes called EIF2 and EIF2B. Next, please, is a build. That in normal situations, these two enzymes work together to synthesize the proteins that are required to carry out the everyday activities of cells. And these normal proteins are depicted in this cartoon by the little squiggly yellow lines at the bottom of the cartoon. Next, please. But when cells encounter various types of biological stress, what happens is that one of these enzymes, EIF2, gets phosphorylated. It changes shape. And when this happens, it no longer works in partnership with EIF2B, but it actually inhibits the activity of EIF2B. And as a result, there's a decrease in normal protein production, and there's an increase in different types of proteins that we call stress proteins, and these are depicted by the slightly different shape here, the red triangles. Next. And in addition, when the ISR is activated, these stress granules form that contain a protein called TDP-43. And many of you have probably heard about TDP-43. It's been reported in the literature quite often that TDP-43 is a protein that may be involved in the cause of ALS. Next. And in conditions where where there's a chronic exposure to stress, what we believe happens is that there's an accumulation of these stress proteins and also an opportunity for TDP-43 and these stress granules to further aggregate into these TDP-43 aggregates that, in conjunction with the increase in stress proteins and the reduction of normal proteins, can lead to toxicity. It can lead to the eventual death of the cells that express the ISR. Next slide, please. And we believe that this pathway, the ISR, is intimately involved in the cause of ALS because TDP-43 aggregates have been found in the vast majority of cases of ALS. Next slide, please. So we and others have tried to find ways to inhibit the ISR as a potential treatment for ALS. Next. And the first major breakthrough was the discovery of a small molecule called ISRIB, and this was discovered by our own Carmela Sidrowski while she was working at UCSF, and now, as I mentioned previously, she's now with Calico as the head of research. And what she discovered was that the way that ISRIB inhibits the ISR is by binding to one of these two key enzymes, EIF2B. And when this molecule binds to EIF2B, it stabilizes the formation of EIF2B and also changes the shape such that it becomes less sensitive to the effects of stress. Next slide, please. So we generated a series of small molecules based on ISRIB, and we call these collectively EIF2B activators. And we've tested these molecules in various preclinical model systems to see whether or not these 2B activators could impact the ISR. The first set of experiments I want to describe to you are experiments conducted essentially in a petri dish where we've taken neurons and expose them to various types of biological stressors. And when we expose these neurons to stress, we see the formation of these TDP-43 containing stress granules. And you can see them in the red circle in the cartoon on the left, and also in the first microscope image of one of these neurons that have been exposed to stress. And when these cells are exposed to, then if they're treated with 2B activators, we can prevent the formation of these stress granules. In addition, we can also quickly dissolve stress granules that are formed in these neurons. Next slide, please. So besides these experiments done in a dish, we wanted to see whether or not these 2B activators could impact the biology in a living organism. And so for these experiments, we generated a mouse that contains a mutation that encodes the EIF2B enzyme. And we know that this mouse model does have a chronic activation of the ISR. So this is a great model system to see whether or not these 2B activators can impact the biology in a living organism that has a chronically activated ISR. So we discovered that this mouse model has some changes in the structure of the nervous system, including the brain and spinal cord. And this mouse model also has some changes in their behavior, and these mice have some problems with their ability to walk normally, and they have some coordination deficits. So the first set of experiments I'll describe to you are the experiments to see whether or not these 2B activators can have an impact on the structural changes that occur in these mice. So in these images that you see, these are images through the spinal cord of this mouse. And the leftmost image shows what a normal mouse looks like that's been stained with a blue stain that stains a particular cell type in the spinal cord called white matter cells. And you can see that in the normal mouse, there's a healthy amount of white matter cells, but compared to that, the mouse that contains the mutated form of EIF2B, where the ISR is on, there's a reduction in these white matter cells. And if these mutant mice are treated with 2B activating compounds, we can see that there's a prevention of the reorganization of the spinal cord, and so that you can preserve the normal appearance of these white matter cells. Next slide, please. And so I also mentioned that these mice have some problems with their motor coordination, and we tested the ability of these 2B activators to help with their function and balance by putting the mice onto a balance beam, and we can measure the amount of time it takes for the mice to cross this balance beam. So this graph shows the time to cross the balance beam on the vertical axis, and it goes from 0 to 40 seconds. And then on the horizontal axis, we have the age of the mice in this experiment. And we're testing the mice between 24 and 35 weeks of age, which is essentially like the adult lifespan of a mouse. And the normal mice can cross this balance beam in about 5 seconds, and that's depicted by the black line at the very bottom of the graph. And you can see that throughout the course of this experiment, they can cross the balance beam fairly well, 5 seconds throughout their lifetime. But if you compare that to the mice that have chronic ISR, starting at about 24 weeks of age, they seem to have a slowness in their ability to cross the balance beam, and that gets worse as the mice age. And by the time they're 35 weeks old, you can see that it takes them significantly longer to cross that balance beam, maybe 30 seconds. But the mice that are treated with 2B activator compounds seem to be able to retain their ability to cross that balance beam. And by the end of this experiment at 35 weeks, they're able to cross this balance beam in nearly the same amount of time that normal mice can cross. So we were encouraged by these preclinical data, and there are other preclinical data that we've generated, but for the sake of time today, I won't present everything. Next slide, please. But these data were encouraging enough for us to advance our program, the lead molecule, which is called ABBV-CLS7262, into the clinic. And over the next few slides, I will go through some of our clinical experiences with the lead molecule. But before going there, I thought maybe I'd just quickly recap the mechanism here on the next slide. In this cartoon figure, I think you've seen this already, that shows what we think is happening in ALS and in chronic conditions of being exposed to stress, that there's an accumulation of stress proteins and TDP-43 aggregates. Next, please. And when 2B activators like 7262 are added to this condition, what this drug does is that it can alter the ISR by doing several things. First, it can help restore normal protein production in these stressed nerve cells. It can reduce the accumulation of stress proteins that may be toxic to the cells. And then finally, it can dissolve the TDP-43 containing stress granules. All right, next slide, please. So now let me just recap what our experience has been in the clinic. We first started our clinical development program in healthy volunteers. Next slide. And in these phase 1 studies, so far we've dosed over 100 healthy volunteers with the molecule 7262. And what we learned is that this drug can be administered by mouth once a day. The side effects or adverse events were all non-serious, and they were mild to moderate in severity. We know that the drug is having its intended pharmacological effect by measuring the activity of EIF2B and also suppression of the ISR in the blood cells that are collected in these healthy volunteers that were part of this trial. And the next slide shows you some of these data. We do know that we have drug in the part of the body where it needs to go. And we verified this by collecting cerebral spinal fluid, which is the fluid that surrounds the brain and spinal cord, and measure the concentration of drug there. And we did measure that at the higher doses, we did have sufficient concentrations of drug that we believe will activate EIF2B in the brain. Okay, next slide, please. These are the results of the experiments that I mentioned, where we verified the pharmacological activity of the drug in the blood cells from our healthy volunteers. So as I mentioned before, EIF2 and EIF2B work together to synthesize normal proteins. Next, please. And this protein production requires energy, and the energy comes from chemical energy that's stored in a molecule called GTP. And these two enzymes work to release this energy from GTP, converting that molecule into GDP. And this constant cycling of GTP to GDP that produces the continuous energy that's required for protein synthesis. And we can measure the cycling of GTP to GDP in these blood cells from our volunteers. Next slide, please. And this is a summary of those experiments. The green bars depict the conditions where people were given active drug, 7262, and the gray bar are people who didn't get the drug. And you can see that at 6 and 24 hours after dosing 7262, you can see an increase in the cycling between GTP and GDP. So we are affecting the activity of EIF2B in these blood cells. Next slide, please. We also show that we can suppress the ISR, and we measured one of those stress proteins called TRIB3, And again, at 6 and 24 hours after dosing 7262, we can see this long-lasting suppression of the ISR gene protein, or the gene that encodes the protein TRIB3. Next slide, please. So based on the data that we collected in healthy volunteers, we did advance our program into people with ALS. And so we started a small study in people with ALS. Next slide, please. And this is an ongoing study. We enrolled 31 people, and the first people who entered the trial have been in the study for over a year now. And so we've been continuously collecting the side effects from this study. And the most common side effects that have been observed to date include the ones listed here, headache being the most common, followed by nausea, itchiness, constipation, and dizziness. But we were encouraged by the safety profile from this study, as well as our healthy volunteer studies. And next slide, please. We are now part of the HEALEY platform, as you know, as Regimen F. And our regimen is designed very similar to the other regimens in the platform to date. There's an initial 6-month randomized control period where people can get either active drug or placebo. And the enrollees are 3 times more likely to get active drug than placebo. And if you are randomly selected to get the active drug, you will be assigned to one of 2 different dose levels. There's a lower dose and a higher dose. And both of these are, it's very important to point out here that both of these drug doses are expected to be sufficient to activate eIF2B in the brain. Following the 6-month randomized control period, people can move into an active treatment extension or an open label extension. And I just want to make a statement here because we've heard that there is some concern among the ALS community that some of the companies, smaller companies in the platform may not be able to fully support this active treatment extension. But because of the way that we're funded and our partnership with AbbVie, I can confidently say that we will be able to honor the obligations that are part of this protocol. And this is as long as the drug is continuing to be developed, as long as it doesn't stop because there's a safety concern or because there's clear evidence that the drug doesn't work. As long as these conditions aren't met, we will be able to support the active treatment extension. Next slide, please. So I just want to quickly recap the refresher, next slide, and tell you why we're so enthusiastic about 7262 being evaluated as the Regimen F in the platform. Next, there's a series of builds here. We know that the ISR is activated in ALS, and our molecule is a potent inhibitor of the ISR. And we know that this it inhibits the ISR by binding to and activating eIF2B. TDP-43 aggregates, we believe, are observed in most cases of ALS, and therefore it's fundamental in the pathobiology of ALS. And we've demonstrated that 7262 can dissolve these stress granules that contain TDP-43. Next slide, please. We verified that the drug is having its intended pharmacological effect in humans, and this is from those experiments conducted in the blood cells collected from our volunteers. Next slide. And we think we've identified the right doses to be explored in this trial. And we did this by measuring the CSF, the concentrations in CSF, and we verified that the drug levels are sufficient to activate eIF2B in the brain. And unfortunately, our understanding of the biology of ALS is still incomplete, and we're hoping to contribute to the knowledge about ALS by collecting body fluids during this trial, which include blood and CSF. And I think this will really help to improve our understanding of ALS, and more importantly for us, the role of the ISR in ALS, and hopefully lead to the identification of those people who are more likely to respond in future trials with 7262. So that was it by way of this refresher, and I think we have a few minutes left, and so we're happy to answer questions from the audience about our program.
Thank you so much. This was really a great overview. And as always, we have lots of questions. So I'm hoping that you and your team can take them. As always, we have a very attentive audience and have lots of great questions about the mechanism and other aspects of the trial. So to start, the first question is, you spoke about the mechanism that you're targeting, the ISR. Is that relevant for familial or genetically based ALS as well?
Yeah, we have no reason to suspect that the ISR, the activation of the ISR will be different in sporadic versus familial forms. And so our enrollment is open to any forms of ALS. Great.
And then again, you showed some great preclinical data, and in particular, you were talking about the biological stressors in vitro in different experiments. What biological stress would there be in humans, in people living with ALS, that cause the ISR to be abnormally activated?
Yeah, that's a really great question. And, I think it all comes from our lack of understanding of what causes ALS. I mean, there are many different putative causes that can trigger ALS, and the types of stressors that activate the ISR can come from nutrient deprivation, certain types of infections. I guess, there are a variety of stressors that could impact the ISR through various sort of signaling kinases that coalesce on EIF2. And accumulation of unfolded proteins in the endoplasmic reticulum. I mean, there are a number of different types of biological stress. And so we think that this mechanism might be sort of a final common pathway that could be improved with this mechanism. I don't know if Carmela might want to add to that, our ISR expert.
Yeah. Oh, sure. I think you mentioned a lot of the physiological stressors. There's this function of two important cellular organelles, both the endoplasmic reticulum and the mitochondria will turn the pathway on, as well as, changes in metabolism, starvation, hypoxia, lack of oxygen. So it is really a common mechanism downstream of a lot of these physiological stressors that we are modulating.
Great. There's a few more questions about the mechanism of action. So in some of your graphs, you showed some, the time course of response to your drug. And potentially in some of the preclinical studies, the response to the drug was rapid. What type of time frame are you expecting in terms of positive response to the drug in people living with ALS?
Wow. That's a big question, and I think it's very hard to predict. We know that ALS is it's a neurodegenerative disease that takes months to unfold, possibly years, and so I think we'll have to wait for that sort of period of time before we see noticeable improvements in the clinical aspects of ALS. I mean, some of the preclinical results can happen very quickly, but it may take some time for those to get translated into hopefully clinical benefit. And I think we have to take be a little bit cautious in terms of translating mouse timescales to human timescales because their lifespan is quite different. So I guess I don't have an exact answer to this, but I think we'll have to let the trial run its course over the course of several months to possibly a year before we see a noticeable clinical improvement.
Yeah. And there's more questions about sort of the translation of mouse data to humans. So for example, can you comment again on the graph where you were showing motor function in mice in response to your drug? There seemed to be a natural improvement, not just slowing down of disease. So would you expect the same response in people with ALS?
Yeah, I want to be a little bit cautious about this. I guess, it looks like from that graph, there might be a little bit of improvement. But I think it would be hard to, I think it would be great if we were able to demonstrate this kind of an effect in humans, but I think maybe we shouldn't set our hopes too high on that. But I would say that based on the mouse data, we could hope for hopefully a stabilization of progression. And I think that's what the mouse data really shows.
Do you expect any changes on brain MRI?
Well, we're not measuring it. I guess it's hard for me to say what we could expect. So in some of our mouse experiments, we did see some changes in MRI that were in the positive direction. But since we're not measuring it in the clinical trials, I don't know whether or not we can expect to see it.
One more question about mechanism, and then I'll take a few questions about trial design. There's a question about the target of your intervention, which is ultimately TDP-43. So if you improve or kind of reduce the accumulation of TDP-43, do you expect mobility to improve? Again, do you expect an effect on function in the legs?
Yeah, I think it's similar to the question about the graph in the mice, that I think what we are hoping to see would be a stabilization at best of disease progression. But I think it would be maybe a little bit too optimistic to expect to see a dramatic improvement in function, maybe slightly, but I wouldn't go so far to say that we'll see dramatic improvements.
The question about trial design. So you explain how the trial has a placebo-controlled randomized phase, then followed by an active treatment extension that you're committed to. So is the active treatment extension only for people who were part of the randomized phase, or would that be offered separately?
Yeah, the way that the protocol is designed is that people who were part of the randomized control period can then go on to continue to receive drug in the active treatment phase of the program.
There's a couple of other questions that are about the same topic, so I'm going to lump them together. And a couple of people are asking about expanded access, and perhaps can you help us understand the difference between what you just presented and the possibility perhaps of an expanded access in the future, which is a different, it's not to be confused with active treatment extension. I just want to be very clear that the nomenclature can be a little bit confusing, but we're talking of something different. So again, the active treatment extension is an integral part of the study after the randomized phase. And then, the separate question is, will you consider at some point, and if so, when, or what are the factors that will have to decide whether to open a separate expanded access protocol?
Yes, this is something that is under consideration, and maybe I'll let Emmanuel address this one.
Yeah, thank you, Sabrina, and everybody for asking this very important question. So since ABBV-CLS-7262 is a new investigational drug with, as you heard, with a new mechanism of action and is not yet approved in any countries, so its use is currently restricted to clinical trials where we can monitor the safety of each participant closely. And at this point in time, even though 7262 appears to be well tolerated in healthy volunteers and people with ALS, we are still collecting incremental safety information. But having said that, we are considering a limited EAP, so expanded access program, no sooner after the first futility analyses will take place in the HEALEY platform. And once the results also of the placebo-controlled portion of the HEALEY platform trial are going to become available for our regimen, the Regimen F, depending on these results, a larger EAP may be initiated. And so just to be very clear, in accordance with the applicable guidance, the EAP will be intended for people with ALS who do not qualify for a clinical trial in which ABBV-CLS-7262 is tested.
Right, so it's a different program
Exactly.
So there's a couple of questions, and again, I'm going to lump them together about your phase 1 study. A couple of questions about, what results you've seen. Can you tell us more about the other study?
Yeah, it's, we can't talk too much about it since it is an ongoing trial. With respect to safety information, the safety data are being continuously monitored by a safety monitoring committee that has not made any alterations, has not suggested any alterations to the conduct of the trial, which is good news to us. And just so people know, the safety data that are periodically being collected from that ongoing phase 1b trial are then transmitted to the HEALEY DSMB. And so that committee that oversees the safety for the HEALEY platform does have access to the safety information that is coming from the ongoing phase 1b trial. So data will be exchanged between these safety committees, but I have nothing, I guess, to report at this time that I can put into slides.
Right. So I think we took all of the questions, and while we wait a few more minutes just to make sure that people don't have any last minute question, I just wanted to give you, Bill, and your team the opportunity to add anything that you may want to add, about Regimen F.
I mean, maybe others on the team will have additional comments, but I'm very grateful to the people who have enrolled to date. I don't know if you are on the call or not, or their families. Thank you for your participation in this and other trials. And thank you to Sabrina for allowing us to be part of this very important trial.
Thank you so much. And indeed, there's a few people who are part of the trial who are on the call. And again, they sent a few messages. So I'm really happy that, people are saying that this work provides them with hope, and they're really grateful for all the work that you are doing to really develop this drug. So we are equally excited and very happy that the Regimen F is going well and is enrolling well. There's a few last minute questions, so I'll just ask you before we really close the meeting. So can you tell us more about the design of the study? Couple of questions, I'll put them together. What dose, what type of, do you have different dosages in the program, and how long do you expect that you will take for people to notice an effect and impact on their disease?
Well, the phase 1 healthy volunteers study explored a wide range of doses, and that is to really carve out a good safety profile across a wide range of potential doses. In that smaller phase 1b study, we did explore a series of doses that we think that all three of them, I think, are going to be efficacious. Predicted to be efficacious. And in the HEALEY platform trial, we've narrowed that down to 2 doses. The lower those 2 doses is our main dose. We believe that will be fully saturating the target. We did add an exploratory higher dose for some additional biomarker analyses and to see whether or not there might be a safety concern. But again, to reiterate, both of those doses we predict will be saturating the target. There was another question about.
Yeah. How long does it take for people in the trial to experience some effects from the drug? Yeah,
I mean, that's hard to predict at this point. I mean, it's if there's no improvement, then essentially, what we're expecting is a slowing of progression. So, I want to be somewhat realistic here that people in the trial will probably experience ALS similar to the way that they've been experiencing it to date. And it's really hard to detect a slowing of this decline, but that's really what we're expecting to see if this drug is efficacious. So it's going to be hard to notice that.
Yeah, there's a couple more questions. We often see this on the webinar that we have, last minute questions, all great questions. And one is about the approach of using cocktails of drugs that we often talk about in ALS, because you spoke about stress. Is there any and do you have any suggestions in terms of combining your drug with other drugs that also target the same or similar pathways, like other drugs that target stress, for example?
Yeah, I mean, I think that's a great question, and it's still an unanswered question. And I think it'll really depend on how neurologists in the future will treat ALS, whether or not it will be by combinations of multiple types of drugs, or if a single drug will be selected. I think the jury's still out on how ALS will ultimately be treated, but I can imagine a future where there will be combinations of drugs. I don't know, Sabrina, if you yeah, sorry.
No, I think it's absolutely, I have nothing to add. I completely agree with what you said. It's a great question. I'm so glad you're on the call because I wouldn't know how to answer this one. Have you used AI to develop your drug?
Yeah, that's an interesting question. I can't think of a situation where AI has explicitly been used to develop this drug or the program. I don't know if anybody else has anything to say about that one from the team here at Calico. No, I guess not. So yeah.
Yeah. So thank you so much again. I think we are really always great to have you on the call. And I have to say that your previous webinar, when you gave a longer overview of your drug, was the most viewed webinar. I would call it a viral video that was viewed by 1,500 and more than 1,500 people. So thank you so much for providing all this information to us and for coming back again, because we have people who really wanted to talk to you again and ask you questions. So I suspect we may invite you again if you don't mind. Really, it's always great to learn from you. I would invite everyone to continue to follow this webinar series. Next week, we'll make sure to provide with the links to all the recordings, all the great brochures that Catherine and Alison have prepared. We have lots of information on our website about this regimen because we're very excited about it. And we have a lot of information on our website also in terms of which sites are active. We have plenty of spots, literally dozens of sites across the country, hundreds of spots for participants. So we would encourage everyone to consider enrolling and really, because the science, as you've heard, the science supporting this drug is really phenomenal. So hopefully we'll get the enrollment completed very soon so that we can then look at the results and tell you more about those. So
right.
Thank you so
for
much.
the invitation back.
Everyone.
Yeah, nice speaking to you all.
Thank you.
All right.
Thank you. Thank you so much. Bye-bye.
Bye.